Hi, we made it to chapter 10 and we're talking about proteins now. Proteins is one of the coolest chapters, I think. So we're going to go over amino acids. It's where we got to start, starting at the smallest portion and then getting into protein formation. All right, so we're getting bigger. All right, we're talking about whole proteins in 10.2. And then we're talking about the three-dimensional structure of those proteins. All right, so we're going to get through sections 1, 2, and 3, maybe just section 10.1 on this video. So we're talking about amino acids, and they're called the building blocks of proteins. All right, so they're one of the major building blocks of life, right? Amino acids, you've probably heard of them before. So proteins are polymers of those amino acids. So I said we were starting small, right? So those are just the tiny pieces, right, of the proteins. So those amino acids are going to make up what proteins are. All right, so the very order of those amino acids and the structures that they create, and then therefore the functions that they relay as proteins. So proteins are polymers of amino acids, and there are 20 commonly occurring amino acids, and proteins contain as few as 50 or even thousands of amino acids. We can be smaller as far as a string of amino acids is concerned, and then we're going to talk about something called a peptide. But if we get to 50 amino acids or so, that's where we're going to start referring to these polymers as proteins. But once again, they can get to be very large. So the order of those amino acids is really important, as I was saying. If you change the order of the amino acids, you're not going to have the same structure, right? The biochemistry is going to change, right? The attractions between amino acids is going to change then, and the structure is different. Therefore, the function is going to be different. Function is all about structure, all right? So function is going to be a factor of structure or vice versa. So proteins can do all sorts of things, all right? They're really important, right? It's what differentiates us from an insect crawling on the ground. Our proteins, right? They're amazing. So what are some of the things that proteins can do? Well, like transport oxygen in blood, for example. All depending on what proteins we have, it makes us what we are. They are part of the main components for skin and muscle. All right, there's more there than just protein, but protein is essential, and we need proteins. We need to secrete certain proteins to signal to our body that we are under a microbial attack, for example, so to signal against infection. So there are enzymes that you need for your body to carry out its daily functions. Those are proteins. Also, to regulate your hormones, you've got proteins that do these really important jobs. So, and look, I already went in and fixed these slides this time. Amino indicates a protonated amine. So a protonated amine. So you guys, y'all remember we talked about these things before, right? So whenever we're talking about a protonated amine, right, and we're talking about a protonated amine, we're talking about the N-terminus, all right, of a protein. So you're going to hear about this a lot. And so amino acid, what does the acid mean? All right, that's referring to a carboxylic acid. Now remember those functional groups? We talked about that before. So before, that means carboxylate, right? So we're talking about conjugate base of a carboxylic acid. So whenever you're talking about this acid part or this conjugate base of a carboxylic acid that we're going to have on these amino acids, this is going to be referred to as the C-terminus, right? So you're going to be able to identify the N-terminus and the C-terminus. Okay. And all of these amino acids are going to be bound to a central carbon that's going to be called the alpha carbon. All right. So you're going to have the alpha amino and alpha carboxylate groups that are attached there. All right. And the alpha carbon is going to be bound also to a hydrogen atom. All right. And a side chain. So y'all remember whenever we talked about chiral carbons, right? So, here we've got this N-terminus, we've got this protonated amine, we've got this carboxylate group, and we've got a hydrogen. So we've got 1, 2, 3, and the thing that's going to make all of the amino acids unique is this R group right here. So that's the side chain of the amino acid. And you can go ahead and, even though you're going to have this on your best friend packet, right? So you want to take out your best friend packet whenever you're watching this video. So you're going to know what the amino acids are and what the structures are. But if you can learn this, it's really going to help you to test better. I would be able to draw it if I were you. So if you can draw this basic structure, all right, just basically the backbone, everything is going to stay the same for a lot of these except for, all right, that R group. All right. Well, for all of them, the R group is what's going to be different. All right. We're just going to have one amino acid that's not going to be exactly chiral, and we'll talk about that. But that's what you're going to be able to do, is know what the backbone is, be able to draw what I've put in purple, and then you're going to notice what's different, and what makes all of these amino acids different is this R group, the side chain. Okay. So that alpha carbon, you remember, is a chiral center most of the time. And see, I told you I would tell you, glycine is going to be a little bit different, okay, because you have 2 hydrogens. So where there would be an R group for most of those amino acids, glycine has a hydrogen. And if you have 2 of the same thing, you can't be chiral, right? So what we need in our body are L amino acids, right? So we need the L amino acids. Those are the building blocks of the proteins that we have, that we need. So we do have some D amino acids, but that's not what's essential to us in our life, really. So remember, D sugars, L amino acids. So L amino acids, that's what we need, and we need the D sugars, right? So be able to remember those differences, right? Since you're taking a cumulative final. So that side chain, all right, or that R group, that's what's going to be unique. That gives that amino acid its characteristics, right? It's going to affect polarity and, different things like that. All of these interactions are going to be determined by what happens in that R group, what's unique about that R group. So 20 amino acids, the 20 amino acids that's going to be found in most proteins. So there are these families of organic compounds, right, where we have alkanes, aromatic sulfides, alcohols, phenols, thiols, amides, carboxylates, protonated amines. You've heard of all these things. You've already heard of these things. So they can all be represented in those amino acids, right? Or in the proteins. So you're going to see, if you look at your best friend packet, exactly what I'm talking about. So those are going to be parts of a lot of those. So the properties of those functional groups is going to affect polarity, as I just said, and whether you're going to have amino acids that might be neutral, acidic, basic, okay? Really important in regards to folding and structure and function. So if I were you, I would make a table that kind of condenses all of this, or we might do this in class in person. We'll make a table together to kind of help organize this in your thoughts. You're going to have to know that some amino acids are nonpolar. So remember polarity and remember those elements and what's going to make a structure polar or nonpolar. So with glycine, overall, we're going to have something here that's nonpolar. Okay, so you look at those charges, you have a +1 and a -1, all right, 0. So you've got something there that is actually balances out to a 0 charge, right? So, on just a second. All right, so we've got glycine, and we've already talked about glycine being different as far as that chiral carbon goes, because it's not chiral. You've got 2 hydrogens. And then, so what I said before, remember this backbone. Remember the overall structure that stays the same for all of them, and just note what's different about each one. Just be able to identify, like alanine, for example, by that side chain, that R group. So you have here this hydrocarbon, right? You've got a carbon and you've got 3 hydrogens right there that's attached to that chiral carbon. So this is alanine, and you'll notice that all of these have one whole name, a 3-letter abbreviation, and a 1-letter abbreviation, and you're going to have to know those. Okay. I think that's on your best friend packet, but it might not be. Just go ahead and I would learn these, even if they are on your best friend packet. If they're not on your best friend packet, well, you have to start memorizing these, right? So if you look at valine, see, look, that one kind of looks like a V for valine. Try to think of little ways to help you remember those side chains, okay? And so you see that the abbreviation is usually the first 3 letters. That's not always true, because there'd be too much repetition and it just doesn't occur that way. And then we have the 1-letter abbreviations, which are sometimes the first letter of the word, but not always. Then we have leucine. Okay, so remember leucine, that's just a little different from valine. So, you know, think of ways to remember this. Like you can think of them in a certain order, like leucine comes after valine. And then if you've got isoleucine, all right, then you'll know that that's also a little bit different from leucine, right? Just take a look at that side chain. And where you've got phenylalanine, all right, think like phenolic, think of what some of those things kind of look like. You've got an aromatic group there, all right? So start studying these, start studying them, okay? So these are nonpolar amino acids, and that's going to continue on the next slide. So we've got methionine, all right, methionine. So we've got a sulfide here that makes methionine a little bit different. And we've got proline, okay? So proline kind of looks like a backwards P a little bit. And then tryptophan, all right, tryptophan. So we've got something aromatic here, an aromatic group. So you remember that, like tryptophan. You ever heard of that, like being the thing that makes you tired from eating too much turkey, all right? And then maybe a turkey smells good. It's aromatic. It's got an aroma. So I don't know. However you can think of to memorize these, go ahead and stick with that. But these are nonpolar amino acids. These are going to be the nonpolar amino acids. So think about what's going on there, all right? So yes, you have like a nitrogen right here on tryptophan, and you have a carbon and everything, but the electronegativity differences there are not so great so that this is going to have some overall polarity. It's still going to be nonpolar. And the same thing with carbon and sulfur, okay, and methionine. So these are the nonpolar amino acids. Then you're going to have polar amino acids. So see, it's good to make yourself a little table where maybe you don't have all these ball and stick models and you can just kind of go through, see it all in one eye shot. So you're going to have primary alcohol. So remember, we went over primary, secondary alcohol, so on and so forth. So here you've got a primary alcohol. So remember, you're looking at what's attached to the carbon that's attached to the alcohol, right? So you've got a primary alcohol right here with serine, okay? So you've got you've got some polarity here, all right? Now you've got a secondary alcohol. So of course, this is also going to be polar, and that's threonine. And with tyrosine, you've got a phenol and you've got this hydroxyl, okay? So that's going to be tyrosine. And notice, tyrosine is going to have a Y as the letter. So it's going to be the second letter of tyrosine is going to be its one letter abbreviation. And for threonine is T. So see why we can't all just be the first letter of each name of an amino acid. So cysteine is going to have a thiol, right? So you see that there? You see that? Cysteine is CYS, and the one letter abbreviation is going to be C. Okay, so these are some polar amino acids, and here neutral. Okay, so polar amino acids that are neutral are going to also include asparagine. Okay, so we see that we've got this polarity here. If we look at our if we look at our elements and try to understand like what's the electronegativity here and what's the polarity. So we have an amide, all right? We have this amide, and that's going to make asparagine. So you're going to have ASN and N, all right, as the abbreviations for asparagine. And note that this is a polar neutral amino acid. But if we have a polar acidic amino acid, all right, like we've got asparagine there, and look, just slightly different here, all right? We've got this carboxylate now on aspartate. So remember when we talked about naming acids and all? So asparagine, all right, is where we have this amide. Then we change this just a little bit, this side group, and now it's a carboxylate. That's aspartate. So however you can think to remember it. And so glutamine, we have a polar amino acid neutral glutamine, and then change that just so you have carboxylate, that's glutamate, glutamate. So we have some basic polar amino acids also. So that would be histidine, okay? So get a good look, all right, at that side chain. All right, look what you've got here. And if you've got a protonated amine here with lysine, this is a basic polar amino acid. And also you've got arginine, okay? So just start studying these. So you notice that some of these amino acids had an asterisk. Those are the essential amino acids. We cannot make those in our bodies. We have to get those by consumption. And two of those amino acids that are arginine and histidine are needed for children, but not adults. So proteins that have all of the essential amino acids are known as complete proteins. So remember, you need to know vocabulary. Those are known as complete proteins. So those are like soybeans and eggs, milk, fish, poultry, right? All of these things are complete proteins. And some plant sources, all right, are they're going to give incomplete proteins. So they're not going to have all of the essential amino acids in, every protein that's in every plant. So you're going to have to kind of balance and say, okay, well, I need these proteins, and I know that these complete proteins are in these plants, or whatever, these proteins are in these plants, and so on and so forth, so that you make sure that you get all of the proteins that you need. If you're a vegetarian, for example, if you're just eating plants, you need to make sure that you are getting all of the proteins that you need. So you can get a complete protein meal by combining different vegetables, different plants, vegetables and fruits, so on and so forth. So You just have to be really careful that you're not missing out on anything in your diet. Okay. So we need to classify our amino acids, right? And we talked a little bit about that already in polarity and charge. So, or as far as acidity and basic qualities are needed to be known. And amino acids are going to be separated into two broad classifications based on the side chains, mostly nonpolar and polar. So make sure you know that, and you put a little star by that. Make sure you understand polarity. You're really going to have to know it. So with a few exceptions, those side chains that are nonpolar, they're going to make for a nonpolar amino acid, are usually hydrocarbons. And that's going to be hydrophobic, right? So you're going to have nonpolar, hydrophobic. Okay. And tryptophan has one nitrogen atom, all right? But that just contributes a little bit to the polarity. And we talked about that whenever we were looking at that just now. It's because of the electronegativity difference. So altogether, tryptophan ends up being nonpolar. And similarly with methionine. So those were a few exceptions to that hydrocarbon rule as far as non-polar amino acids go. So the polar amino acids are going to have three groups that are going to be neutral, acidic, and basic. So the polar amino acid side chains, they're going to have they're going to be functional groups that have an uneven distribution of those electrons. So that's going to affect the way that string of amino acids that have those functional groups behave, what kind of interactions they can have. So, and attractions, right? Attractions, interactions. So all polar amino acids, except for cysteine with that SH group, you remember, can form hydrogen bonds with water. So they're hydrophilic, right? Remember, we went through this type of stuff when we talked about solutions. So you should be familiar with this. Polar acidic and polar basic amino acids have side chains that are going to be charged. And so what you're going to have then, because you have these interactions with water and you have these charged side chains, you're going to have ion dipole interactions, right? So you're going to have these pretty strong interactions because of these amino acids whenever they're present in a protein. So we're going to have those abbreviations probably on your test, so you need to know those. So the amino acids are going to have 3-letter and 1-letter abbreviations. So 20 of them, of the 20, 11 have a 1-letter abbreviation that start with the first letter of the name of that amino acid. So that's really easy to remember. But then there's 9 that have 1-letter abbreviations that are not the first letter. So you have to be careful. So make sure you're looking out and you're trying to learn which letter abbreviation that amino acid is supposed to start with. And we have these abbreviations because if we needed to write out these full names for 1,000 amino acids in a protein, gosh, who wants to do that? It takes forever. So that's why we have these abbreviations. Okay. So, all right, we have this table here that sort of goes through all of these amino acids where you have a rationale for why they're naming something with 1 letter and why that particular letter, I mean, and you're going to have the code for that letter, the 3-letter code for that letter or abbreviation, and it tells you which amino acid that belongs to. So if we look for methionine, for example, we see that the 3 letters are the first 3 letters of the name methionine, and the 1-letter abbreviation is M. If we look at glycine, we see the first three letters or that three-letter abbreviation, and that G is the abbreviation for glycine. So the ones that are the most common are going to get that privilege of having the one-letter abbreviation being the first letter of the name. So now you're going to notice there are these 9 amino acids where you're going to have a different one-letter code from the first letter of the name, because you know it's already used in a lot of these cases. So where you have your arginine, R8, think of it like R, like a pirate, like R, you know, whatever way you can think of to remember it. All right, phenylalanine. All right, so we have proline and we have phenylalanine. So, you know, if you look back, you see that proline gets that P. So phenylalanine, what it sounds like, it starts with an F. So phenylalanine, F. Tyrosine, all right, we've already got T taken. So it's the second letter now, Y, tyrosine. So tryptophan, I don't, tryptophan, you know, however you can remember it, it kind of sounds like a bit the way Elmer Fudd would say tryptophan. So, so that's a W. Aspartate, D, like aspartate, they say. Aspargine, all right, so it has an N there. Glutamate, so it's an E, like glue. Glutamine, all right, Q, like cutamine. And lysine, I'm not exactly sure how cutamine is supposed to make you remember Q, but lysine is a K because K is close to L in the alphabet, right? And we've already used L, and that's leucine. So just get busy looking at these. So amino acids as weak acids. So all of these amino acids have amine and carboxylic acid functional groups, right? So they all, and that's important. So we can have charged and uncharged forms. Remember, like we talked about with drugs in the body and so on. So they have, you know, acid-base forms and ratios depending on the pH, all right? So remember pK and pH, all right? It's not going anywhere. So the amino acid alanine, all right, is shown in the form that predominates at physiological pH. All right, so, all right, physiological pH is about 7.4, right? And they're just giving us one value here. So where we're going to have this form that's going to have no net charge, where we have a protonated amine and a carboxylate, so we've got a plus and a minus, this cancels out, right? So you have a +1 -1 equals 0. So whenever the charges cancel out to a 0 net charge, that's called a Zwitter ion. A Zwitter ion. Okay, so that's a Zwitter ion right there for alanine. So remember, you have to know these definitions because everything is going to build on itself. So each molecule, all right, is going to have a unique pH at which the Zwitter ion is what's going to be present, just the Zwitter ion. So whenever that happens, you are at the isoelectric point, right? The isoelectric point for that amino acid, that molecule. So at the isoelectric point of alanine, the carboxylate ion is going to be balanced by the ammonium ion, all right? So negative and a positive. So the net charge is 0. That's a Zwitter ion, all right? So something that you're going to need to know is when we're working these problems, when you're looking at the carboxylate, you have to know what the pKa is, about 2.3, and the pKa for your ammonium ion is going to be 9.7. So you're going to remember that for whenever we're working our problems. All right. So know that you're going to have forms that are more, that that are going to be, you know, more of the acid or the base form. All right. So we're going to work some problems like that. So the charge on that amino acid, particularly the functional group, is going to be different depending on the pH where that amino acid is located. So whenever the pH is going to be lower than the pI, so whenever the pH is below the pI, then the acid forms predominate. All right. The acid forms predominate. And whenever the pH values are above the isoelectric point, right, the basic forms of the functional groups predominate, right? Basic forms predominate. So make yourself some note cards, okay? Help you remember. Let's work some problems for 10.1, and we'll make a different video for 10.2 and 10.3. So, all right, let's get into a few of these. So we need to do the evens, I believe, and we'll do the odd ones in class. How do we do this? No, we usually do the even ones in the odd ones in the video and the even ones in class. Let's continue to do that. So draw the structure for each of the following amino acids and put an asterisk next to any chiral carbon centers in your structure. Okay, alanine. All right, so let's do this. Why did I write on top of the word? All right, so let's do alanine. So alanine, if you look at any of these tables, the 3-letter abbreviation, that's ALA, and A is going to be the 1-letter abbreviation, okay? So let's go ahead and draw that out. Let's do alanine and lysine, and I think the rest of them, you can figure it out, and you're going to need to use those for practice. So A, alanine. Alanine, repetition doesn't hurt, ALA. So let's draw this out. So we're always going to have this same backbone. All right, that's supposed to be a plus sign. All right, so we've got our carboxylate. So remember, we've got hydrogen and alanine. Remember, right there after glycine, we looked at alanine earlier. So this is going to be non-polar. Remember, we talked about hydrocarbons, and that's going to be hydrophobic then. And so this altogether, this is neutral, right? So just remember what's different about each side chain, and remember the parts that are the same amongst those amino acids whenever you're trying to remember how to identify these, because I know your test might have, this as multiple choice, but it'll be so much easier to just be able to look at these things and recognize what you're looking at instead of, trying so hard to flip back and forth and figure things out with the best friend. And trust me, it'll be better if you just learn it. Okay, so let's do B, lysine. Lysine, that is, remember, we already used L at this point. So lysine, think L, but what's next to L? K. The abbreviation is K and LYS, the first 3 letters. All right, and lysine is going to be polar. Lysine is going to be polar, but let's draw it out so that we can understand that. So how's it like other amino acids? All right, so let's just draw the backbone. Let's draw this backbone. Let's see. All right. So you draw the backbone, and then what have we got for a side chain? Oh, I might need to move this out the way. Oops. Whoops. Okay, that's all right. So let's do another CH2, right? There are 4 of them actually. And then you have your another protonated amine. So here we go. And so you can remember you've got a protonated amine on this one. And this is lysine. All righty. So, and you need to identify the chiral carbon here. We can do that with an asterisk. So it's right here, right? So if you look at those different groups, 1, 2, 3, 4, right? So it's a chiral carbon. That's the one. And the same thing here. That's the chiral carbon. All right, so this is pretty much all you have to do for these kind. Let's see. So basically we've done 10.1 and 10.2 at this point. Okay, so let's see, what else do we have? 10.5, classify each amino acid as polar or non-polar and as hydrophobic or hydrophilic. Oh, did we already do that? Did we already do that? Not all the way, not all the way. Okay, so let's talk about lysine. So lysine is going to be hydrophilic, okay? So we have something that is polar, all right? Right? We do have something that is actually polar. It's basic. Remember, we went over these. And all right, is that all we needed to say? And it's hydrophilic. It's hydrophilic. It's polar, right? It's polar. It can interact with water. It's like saying, hi, hey, water, how's it going? Whereas you have something like alanine that's hydrophobic. It's going to try to get away from water. So this is, you see how these functional groups make all the difference in how parts of amino acid chains are going to interact with each other, how this polymer is going to fold whenever they're in an aqueous environment. All right, so now we've pretty much done 10.5. What about 10.7? Are we going to do this now? Do this in class? We can go over glycine, I think now. Let's do glycine. Or yeah, we can do glycine and histidine. Okay, let's do glycine and histidine. So this is G is glycine, right? Get familiar. I just see G and I just kind of know glycine, but I know you're just starting with this. And HIS, that's histidine. So it's the only one kind of like that. So you probably knew that. And that's an H, histidine. Okay, so let's do A. We want to draw the structure for the amino acid represented by each of the following abbreviations. So we're just going to draw glycine. So let's do our backbone first. Okay, we draw our groups around our chiral carbon, except in this case, it's what? Not a chiral carbon. Boom. That's glycine. That's why glycine is different. Okay. And so this is nonpolar. All right. And that means it's going to be, it's neutral. It's hydrophobic. It's hydrophobic. Okay. So now B. Let's do histidine. So let's draw out our backbone. So you see what's happening every time we're drawing one of these amino acids, we have this backbone where we have this N terminus and we have the C terminus, right? And that's going to stay true for a chain of amino acids that we're going to have something called a peptide, and in proteins we're going to have N terminus and C terminus on the ends. So you're going to be able to distinguish where certain amino acids are in those chains by where you find an N-terminus and a C-terminus in the backbone. So we're not talking about the side chains, just in the backbones. So you're going to have a carbon, carbon. All right. So get to know how to draw these things. So now we've got histidine, where we have CH2, and we've got, let's see if I can do a good job drawing. Oops, nope. There we go. Oops, somehow this didn't come out right. Oh, there we go. Okay, now it's going to come out right. Close. Terrible artist today. Okay. And all right. So we have secondary amine. All right, this is polar basic, and it is hydrophilic. Okay, so let's see what else we have. All right, so let's see, we already gave names to the amino acids that we drew in 10.7. All right, so now we need to do 10.11. All right. So isoleucine has the Zwitterion structure shown. Draw the structure and give the net charge of isoleucine that will predominate at the indicated pH values. So the isoelectric point, right, is going to be 6. So we're going to be looking at a pH of 1.86 and 11.5. Okay, so if we have isoleucine, so what's going to happen is, let's write this down. If the pH is below the PI, below the isoelectric point, then you're going to protonate. So that means you're going to add hydrogens, right? And if the pH is above the isoelectric point, you're going to deprotonate. All right, so you're going to remove hydrogens. Okay, so if we have a drop in pH, all right, if we've got a drop in pH, and so then if we draw our backbone, just like it is for isoleucine, for everything, just draw everything kind of exactly the same here so that you can see. But it's essentially the same as what we've been drawing. All right, you're going to have your side chain. All right, and this is isoleucine, like we have leucine, and then we have isoleucine. That's just a little bit different, right? So what you're going to have is whenever the pH, let's say, is 1.8, and it is below the isoelectric point, which is 6, apparently. So the problem is telling us that. So if that is the case, then we need to, so we're dropping the pH, and the pH is 1.8. What we have to do is protonate. So what you're going to do is you're going to add a hydrogen right there. So you're going to add a hydrogen. So you see what I did? All right. I added a hydrogen. Okay. So you protonate right there. And so now I don't have a negative. I do have this positive. So the net charge now is going to be +1, right? because we have +1 and 0, so +1. So this is at pH 8, all right? And here, this structure where we have our net charge is 0, right? So B, so this was A, and if this is B and the pH is 6, well, our pH then is going to equal our isoelectric point. So just the pH is 6 and the pH is equal to the PI. We don't have to do any more, right? We're done with this problem. Okay. So then what happens if we increase the pH? Increase the pH. If we increase the pH, and let's draw a structure over again. If we have it never hurts to keep practicing and drawing it out. All right. So you're going to have your chain still. CH3. Not a great artist today. Oops. So what we're going to have to do is remove hydrogens, right? We're increasing the pH here. And let's say this is our C, that's 11.5 pH. So what we're going to have to do is remove hydrogens. So look, this is where we have our hydrogens to give. So we're going to remove our hydrogens. So we're not going to be positive. And now we're just going to have NH2, all right? So you see that's where I have changed the amino acid. Okay, and so here, right, so this is going to be what happens for a pH of 9.7 and above, right? So remember, you have to think about the pKa, right? So don't forget, so the pKa, I'll write it here in case you don't remember, is 2.30 for our carboxylate, and for our protonated amine, we have 9.7. Okay, so there you go. That's all you do for this one. Okay, and I think that might be it for that one. Okay, so we're going to do a separate video for 10.2 and 10.3. Thanks.